Welcome to the deep dive. Today we're looking at something pretty remarkable. Profluent Bio's OpenCRISPR-1. This is really a potential game changer in biotech. It's the first gene editor used in human cells that was, well, designed entirely by AI. So our mission today is to unpack that. How did AI go from just crunching data to actually creating a working molecular machine like this?
It's a fundamental shift, really. Traditionally, we found these tools like Cas9 in nature. Bacteria use them for defense. OpenCRISPR-1 wasn't found. It was generated, built from scratch by these large protein language models.
Generated, not discovered. Okay. So how exactly did they teach an AI to do that? What did it learn from all that data?
Well, it learned the language of CRISPR, you could say. They fed the AI models just a massive amount of information. We're talking over 26 terabases of genomic data. That's data from more than 1 million different CRISPR systems found in nature. So the AI basically absorbed the rules, the patterns of how these proteins are built and how they function. Then it could generate totally new ones.
Okay, so it learned the rules and designed a new system. How did it actually perform when they put it to the test in human cells against the standard Cas9?
The performance was pretty impressive right out of the gate. For on-target cutting, getting the job done at the right spot in the DNA, OpenCRISPR-1 was right up there with Cas9. Matched it, sometimes even exceeded it in efficiency and disrupting the target gene.
Okay, so it works well. But the Achilles heel of CRISPR, the big worry, has always been hitting the wrong target. Off-target cuts? Did the AI manage to solve that?
That really seems to be the major leap forward here.
Yes.
It dramatically cut down on those off-target mistakes. We're talking about a reduction of around 95% compared to Cas9.
95%. Wow.
Yeah, the numbers were something like 6.1% off-target cuts for Cas9 in their tests versus only 0.32% for OpenCRISPR-1.
That's a massive difference for safety and potential therapies.
It really is. And there's another safety angle, too.
Oh.
Immunogenicity. Basically, the risk of the patient's immune system attacking the editor. Cas9 has known regions, epitopes, that can trigger an immune response.
Yeah.
The AI design seems to have avoided several of those.
So, potentially less risk of rejection. Okay, that's clever. What about doing more precise things? Not just cutting, but say base editing. Fixing single letters in the DNA code.
Yeah, it handles that too. They modified it slightly, turned it into what's called a nickase. It just snips one strand of the DNA. And when they did that, it could drive A to G base conversions pretty efficiently, rates around 35 to 60%. And what's really interesting is that it worked well even when fused to other AI-generated parts, like some deaminase enzymes they also designed.
So it's AI building the core tool, and AI building the accessory parts too. What about the GPS system, the guide RNA that tells the editor where to go?
They tackled that with AI as well. Separate neural networks were trained just to design optimal single guide RNAs for OpenCRISPR-1. And using those AI design guides boosted the editing efficiency even more. So it's a real end-to-end AI approach.
Right, designing the whole package. And maybe one of the most significant things here is that they didn't lock it up. It's open science.
Exactly. That's a huge decision. Profluent released everything, the DNA sequences, the plasmids to grow it, the source code, under an ethical open license. This really shifts things. It moves gene editing more towards rational, predictable engineering using models rather than just trial and error discovery. It could massively speed up the development of safer, more precise therapies, maybe even personalized ones.
So the big picture here is pretty clear. AI is now stepping up from analysis to actual creation, building biological tools that might be safer and better than what nature evolved. And that brings us to a final thought, something for you to consider. Given that AI can now design these powerful biological components so quickly, and the blueprints are being shared openly, what new ethical questions, what regulatory challenges pop up when this kind of precision genome engineering becomes much easier, much faster, and much more widely available? Something to think about.